Hello, welcome to our seminar series. My name is Valerie Collins. I'm the Applications Manager at RedShiftBio, and today I would like to give you a sneak preview at an application note that we have coming out very soon, titled Characterizing Amyloid-Forming Immunoglobulin G Light and Variable Chains using Microfluidic Modulation Spectroscopy. This is a collaboration that was done with Northeastern University. Here's a list of some of the other articles that we've been happy to collaborate on, and we would like to invite anybody interested in our technology to reach out and see what we can do to help your research. Those publications would not be possible without our global customer base, many of whom collaborated with us early on and adopted MMS in their research labs. All of the work I'll present today was performed at our headquarters demo lab in Burlington, Massachusetts, but we're very happy to announce that we now have 4 demo labs worldwide to make these types of collaborations easier and closer to home. So let's get started into the technology. MMS is an infrared-based technique that can probe protein secondary structure. Shown here are our 4 key pillars of innovation over traditional FTIR. The first one is we are using a high-power quantum cascade laser that's much brighter than the traditional FTIR light source. With brighter light, that means we can go down to lower concentrations and see smaller changes earlier in the process. Next is our fully automated sample handling system. We've implemented the use of a 24 and a 96 well plate to load samples and for complete walk-away automation. We have our proprietary software that is in charge of not only building those protocols to run the walk-away sample analysis, but also to analyze the data afterwards and to produce high-quality figures. And lastly is our high-precision microfluidic flow cell. This gives us extreme reproducibility and allows us to have compatibility across many different buffers. And this is so important. I have an animation on the next slide of how it works. So the flow cell looks like a capital letter Y or an antibody, where in one of the arms of the Y flows in the sample, and in the other arm flows buffer. And every second, we're modulating between sample and reference buffer to get a real-time background subtracted spectrum. This real-time background subtraction means that we're nearly drift-free and have about 98% system repeatability replicate to replicate. So when we see small change, we can be very, very confident in those changes and that it's not something like system noise. With biophysical characterization, there's multiple different levels of structure that you can be probing. So we have primary, secondary, tertiary, and quaternary structure. And what you'll notice from this list is that with each of these levels of structure, we have fully automated and very robust tools available. However, secondary structure, the tools available are circular dichroism, CD, and FTIR, and they have some notable drawbacks. So CD, you're typically at low concentration ranges, and it has poor buffer compatibility, so you will usually have to buffer exchange into a more friendly buffer. FTIR, on the other hand, is typically in the higher concentration ranges, and it suffers from a really manual workflow. So this is where we see ourselves fitting into biophysical characterization, where we offer a fully automated and robust measurement for secondary structure characterization. We are able to provide robust structural information due to our quantum cascade laser, as I mentioned. This gives us 1 wave number resolution and 30 times the sensitivity over FTIR. We are probing the amide-1 band of the IR spectrum. This is dense in backbone conformation information. And then lastly, the real-time background subtraction means that we don't have interference from buffer components like CD, and that we can go in a range of different concentrations. The amide-1 band is rich with protein backbone information because it's detecting the carbonyl stretching vibration, which is highly affected by the alpha helix or beta sheet conformations that proteins can take. It's also sensitive enough to detect the difference between inter and intramolecular beta sheets, which are important for detecting aggregation. Protein structure is critically important to its function and can be useful in detecting changes due to ligand binding, different formulation conditions, aggregation, and things like quality control for batch-to-batch reproducibility. So we've gone over how the system acquires the raw differential absorbance spectrum, but now we'll go through the software and how the software analyzes that data. So we start with the raw differential absorbance, and then the software normalizes that for concentration. So this is how we can analyze multiple different samples, all at different concentrations. We then take a second derivative, and this highlights regions of small change. With the second derivative, we then invert and baseline subtract to get what we call a similarity plot. It's called the similarity plot because we applied the area of overlap calculation to quantitate percent similarity compared to a control. So when I say we have 98% system reproducibility, it's 98% area overlap using this type of analysis. We also use this plot to do our Gaussian curve deconvolution. So this is how we are calculating and quantitating the percent higher order structure of your proteins. All right, so now is the moment we've all been waiting for, where we dive into our collaboration and the data that we were able to generate building off of the recent JMB paper published by Northeastern. The focus of this work is understanding light chain amyloidosis. So we'll start with this cartoon of an IgG, which I'm sure many of you are familiar with. We have two heavy chains colored in green and two light chains colored in blue. And normally, a light chain will pair with its complementary heavy chain and everything will be good. However, in light chain amyloidosis, these light chains could be free in solution, or they could homodimerize with other light chains, misfold, and cause amyloid plaques. These plaques will deposit on various tissues and organs, causing failure across the body. So this is called a form of systemic amyloidosis, and it's one of the most common forms of systemic amyloidosis. Importantly, though, the aggregation mechanism for this is still largely unknown. So the purpose of this work is to look at recombinant light chains and variable chains that are based on patients that are either amyloidosis positive, which will be colored in red, which have multiple myeloma, which will be colored in blue. And these types of samples, these patients are disease prone, but not necessarily disease positive. And then in green is germline, which is non-disease prone. So real quick before I go to the next slide, this whole thing is the light chain, and we'll be focused on the light chain and the variable chain, which is just this top portion here. So in the JMB paper, the authors conclude that the GL samples are much more stable than MM and AL. And so this is one of the figures that supports that conclusion. In the top, in A, we see a CD spectrum looking at the protein secondary structure. So we see it's mostly beta rich, which is expected, with a minimum here around 215 nanometers and a maximum around 200. By monitoring just one wavelength and then doing a heat stress, we can see the thermal unfolding of these samples. And you see that the red line unfolds a little bit earlier than the other two. It's slight but detectable. So this one's a little bit less stable. In the middle here in figure C, they are showing the proteolysis by trypsin across time. So the AL sample is digested very quickly, same with MM, but GL is more protected and is more resistant to degradation. Samples from the 140 hours of degradation are shown here in transmission electron micrographs, and this shows the amyloid fibrils that are formed. And then over here is fluorescence emission by THT across time and agitation. And you can see the highest increase in fluorescence is due to the AL sample, which means that we have the most aggregation in that sample and much lower in MM and GL. Lastly, hydrogen deuterium exchange was performed to see regions of vulnerability and flexibility. So you can see that the AL has the most deuterium exchanged compared to the other two samples. So this one is more vulnerable to the exchange. And importantly, we have this one region, the CDR1 region, that will become important later on. So here's the first set of MMS results showing that we can clearly distinguish between the LC group and the VL group due to the contribution of fractional alpha helix and beta sheet content. The overall LC has a higher fractional contribution of alpha helix, so by measuring the signal at 1656 wave numbers, which is important for alpha helix, we can clearly distinguish the two populations just by seeing the higher amount of alpha helix in the LC samples. Taking a deeper look into the data, though, we'll find in A, this is the absolute absorbance spectrum, B is the second derivative, C is the similarity plot, and D is the quantitated higher order structure bar graph. So clearly these samples have a major peak around 1640 wave numbers, which is known for natively folded beta sheet formation. Importantly, the AL sample has a much smaller peak at 1640, and we can see that quantitatively in that higher order structure bar graph, where the beta sheet for AL is much less than the other two samples. And the amount of intermolecular or aggregated beta sheet is higher in the AL sample, along with the unordered structure. This conclusion agrees well with the hypothesis from the JMB paper that AL is the least structured and can lead to misfolding and eventually aggregated and disease propagation. I wanted to highlight that although CD also does probe secondary structure, it's much harder to quantitate higher order structure. This is partly because the signal for beta sheet is much less intense than alpha helix. So shown here in this figure, the dashed line is 20 mg per mil BSA, which is mostly alpha rich. And the solid lines are 20 mg per mil, same concentration, IgG, mostly beta sheet. And you can see the clear disparity between the intensities of these two peaks. Another difference that makes it difficult to deconvolute and quantitate higher order structure for CD is that a lot of these features are overlapping for alpha helix and for beta sheet. So shown on the right are two figures from this study for the LC and the VL that show a similar trend for LC and VL. But more than that, you can say that it's mostly beta rich, but it's tough to really pinpoint differences in the structure from CD spectra. So this is contrasted by the MMS results that provide the higher order structure bar graphs and table below, shown here, where the LC samples are in the bright colors, the VL samples are in the darker shades, and you can clearly see the difference between each sample. We've compared our results to X-ray crystallography, and there are some notable differences. This may come from the fact that in X-ray crystallography, everything is done in solid state. MMS, everything is done in liquid state. And the conditions under X-ray were done specifically to promote crystallization, whereas with MMS, we're compatible with many different buffer components and excipients. So this can be done under any solution that keeps your protein happy. Overall, the authors showed in the JMB paper that GL is much more stable than MM and AL using thermal unfolding, limited proteolysis, fluorescence via THT, and HDX. We were able to quantitate the structure of those samples using MMS and correlate that structure back to the observed stabilities, where AL sample was the most flexible and the most unordered structure and prone to intermolecular beta sheet aggregation. The authors also go on to pinpoint one of the 8 mutations between AL and GL that occurs in the CDR1 loop. We mentioned that earlier. And with this one mutation, the N32T mutation, you can apply that to GL and greatly increase the aggregation propensity, decrease the stability, and the opposite mutation, if you apply T32N to the AL sample, you greatly increase the stability and decrease the aggregation propensity. So clearly, there's this CDR1 loop is very important for aggregation. And so this is another opportunity to potentially study the secondary structure and see what secondary structure effects are caused by those mutations. Before we wrap up today, I just wanted to briefly talk about our introduction of our second generation instrument, Apollo. We launched Apollo earlier this year to address some of the feedback that we got from our early adopters. One of the biggest hardware changes is that we increased the throughput by implementing a buffer wash station on the end of the 96 well plate. This means that you can wash your samples and wash the cell without taking up space on your well plate. So this leaves more room for samples. Next was our increase in resolution. Our first generation instrument called the AQS3 Pro had 4 wave number resolution, and our new system Apollo has 1 wave number resolution. And importantly, each modulation cycle in AQS3 Pro used to gather 1 data point, whereas in Apollo, each modulation cycle gathers the entire spectrum. So we're saving a ton of time and volume doing the processing with Apollo. This is all while maintaining the data quality. Here I'm showing the AQS3 Pro in dark blue and Apollo data in green overlaid, where you can see that the data quality is maintained across the amide-1 band for both AQS and Apollo. Lastly, I just wanted to highlight the volume savings. Here I'm showing the volume consumed for AQS3 Pro in blue, Apollo in orange across this concentration series from 2 to 100 mg per mil, while maintaining the system repeatability, this line graph above. But overall today, we showed that MMS works really well to help show the whole picture, and it works really complementary with other biophysical techniques like TM, HDS, SEC, things like that. We don't expect to be the one shot for everything. Where a lot of these systems give you a number, like a melting temperature or a size, we give structure. And that can be really helpful in seeing the overall picture and the overall story of what's going on in an aggregation process. So I'd like to thank you for joining us today. If we've piqued your interest, you can find all of our articles and application notes on our website under the resources tab. This application note will be coming out very shortly. And if you have any questions or interest in running samples, please reach out via email. And I believe we have a short Q&A section right after this. So thank you. All right, so I see in chat, the first question is, did you encounter protein instability and aggregation in these samples? Good question. So I believe with the samples that we were doing at like higher concentrations, we may have seen this, we were operating at pretty low concentrations to try and mitigate how much aggregation we have. One of the things to note is that this is a flow system, so we want to avoid clogging our flow cell with aggregates. And so if we do see precipitates, we can do things like centrifuging to get rid of the larger aggregates or filtering to get rid of that. All right, the next question is how much optimization of parameters are needed before measuring each sample spectrum and for each sample set? So the system is very automated in many different ways, and one of them is during the fluid setup, we call that the fluid entry. It performs a lot of those checks behind the scenes, and it will choose the optimum parameters to run. So this is going to be doing it all for you. You don't have to remember any parameters. The next question is, what are the upper and lower protein concentration limits for this technology? Our lower concentration limit is about 0.1 mg per mil. This is pretty low for us. So for good higher order structure, we usually recommend 0.5 mg per mil, but when pushing it, we can go to 0.1. And the higher concentration is 200 mg per mil. It's not necessarily our highest concentration, it's just the highest one we've tested. One of the things to keep in mind is viscosity. At those higher concentrations, you usually have higher viscosity. And being a flow system, we can handle higher viscosity up to about 20 CP. The next question is, are any buffers typically found in protein formulations that are incompatible with MMS? And so most typical buffer components we've tested and we've had very good luck with. Feel free to reach out if you have anything particularly weird, but we can handle kind of low percent organic solvents, which are not typical for buffer formulations, but more like PS80, PS20, sucrose, sodium chloride, histidine, arginine, those types of excipients are all good. And then let's see, next question. Do you demo the system? We do demo the system. So as I mentioned, we have 4 labs worldwide. So you can submit your samples to our headquarters across the globe. And if that looks interesting, then we can bring the system to your lab and see what we can help out with there. All right, so next question is, can the measurement be carried out in low volumes? So we have decreased the volume using Apollo, and we're continuing to work on that. Right now, the volume for Apollo is roughly 800 microliters for triplicate measurements, and that's as low as we can go. If the protein is not beta sheet rich, then is there any benefit to running MMS? Yes. So for even like alpha helix rich proteins to run CD versus MMS, MMS is going to give you a more accurate higher order structure deconvolution. CD will typically overestimate the alpha helical content. But to run other proteins, say if it's in like a formulation buffer that has some excipients, many excipients are incompatible with CD. So our system is much more friendly to other excipients. And in a side-by-side study to look at limit of quantitation, which is the smallest amount of change that is detectable beyond the noise, we did a head-to-head MMS, CD, and FTIR. MMS had about 5 times the sensitivity of CD and about 30 times the sensitivity of FTIR. And that's what I had mentioned earlier with the 30 times sensitivity. All right, what about using lipids or detergents? So we've tested a few detergents. Lipids, it's something that we're learning about. We would love to collaborate and learn more. So lipids, the thing that is really important about doing our measurement is that it's always very clean buffer subtracted. So if your sample is in like say a lipid nanoparticle, I know those can be very heterogeneous, so that can be hard to get a correct buffer match to do that background subtraction. So it would have to be done kind of in pieces, and we'd be happy to collaborate and learn more about that application. The minimum concentration required, so we had mentioned previously, 0.1 mg per mil is our absolute lowest. 0.5 is usually what we recommend for good higher order structure calculations. The presence of some excipients in the formulation buffer can also interfere with measurements. How do you handle it? Yeah, good question. So say in your buffer, you do have some other amino acids, and of course, they're going to have the same carbonyl stretching vibration that we're looking at, or even water has a big absorbance in our region. Because of our real-time background subtraction, those can be very cleanly subtracted because of the way that we modulate. So that does depend on having a very good buffer match. And we've tested this internally to see like how good is necessary. And we can handle like slight buffer mismatches and correct for that and handle that in the software. But overall, to have a clean buffer match would be the best way to handle any kind of excipient that has a signature in the amide 1 band. So there's another question on structure changes in membrane proteins, which must be in lipid vesicles. So yes, that would be difficult. That's something definitely that we want to learn more about, because I think that there's potential there, but it would be very difficult because lipids are typically really transparent in IR. And so that gives us what we kind of call a negative spectrum that you then have to correct for. And if that buffer match, if the lipid isn't perfectly matched in the buffer, then that's very difficult. But we have tested other things in like complicated solutions and been able to tease out the protein information apart from all of the buffer components. Again, thank you so much for joining and asking some really good questions in the chat. And I look forward to hearing from you in the future. All right, so it looks like we're out of questions. If anything pops up, feel free to reach out to email. This webinar will be posted on our website as well, the recording of it. So thank you for spending your last 30 minutes with me, and I hope you have a great day.